you all for coming. Today we have a guest here, Moritz von Stosch. Moritz actually studied here in Aachen as well from 2001 to 2006 and did his diploma engineer in chemical engineering as well. Afterwards, Moritz did his PhD in Porto and did a few of his research stays at MIT and Berkeley as well as Lisbon. After that, Moritz was a lecturer at the University of Newcastle, and he worked for about 3 years at GSK. And at the moment, Moritz is working at DataHow, which is a small startup company based in Zurich, Switzerland. And Moritz's research and work is mostly focusing on hybrid modeling, so the combination of mechanistic and data-driven modeling with applications in bioprocesses. So thank you very much, Moritz, for coming, and the stage is yours.
Perfect. Thank you very much, Arthur, for the very nice introduction, and thank you also for the invitation. It's always a pleasure to come back to the places where I've studied and where I've spent so much time, and somehow give back some of the creative energy that I also experienced during my time at these places. Let me share my screen. Can you see it? Yes, perfect. Okay, I'll minimize just this window here if I can. Okay, perfect. So welcome to this talk on hurdles in the development of dynamic hybrid semi-parametric models and the exploitation for bioprocess development. I'm currently working at DataHow. I'm their Chief Innovation Officer, and as a Chief Innovation Officer, I'm really working between the disciplines. On the one hand side, I'm client focused, and I'm working on client projects where we see client problems and we take on these client problems, then innovate. In particular, we develop new methods and algorithms to address these problems in a very efficient way. And then if we see that there's a bigger market to these kind of algorithms and there are other companies that have the same problems, we essentially pass on the algorithms that we developed in the innovation and research team to the product team, and we formalize it into a software which that can be offered to more clients than the few ones we're working with. And DataHow is a company that is now 3 years old. It's from the RWTH Zurich, a startup, a spin-off essentially. We're all working on the interface between pharma, machine learning, but also chemical engineering in particular. And we have 10 of the biggest pharmaceutical companies already as our customers. We're also working with technology partners such as Siemens, Inforce, or ABB, and we have also several academic collaborations, for instance, with the Technical University of Berlin, and then, of course, with RWTH Zurich. Okay, perfect. So before I start, I really want to say that our vision at DataHow is that we want to use hybrid modeling at the core of everything, because we believe that combining mechanistic knowledge with neural networks or with machine learning in general really gets the best from the two worlds. And by combining the data sources and the knowledge sources, we can increase understanding, we can reduce the requirements on the data side, and in particular, we can excel in extrapolation. And I want to show you in a minute what that means. Hybrid models also enable knowledge transfer. And knowledge transfer is really here from the small scale to the big scale, so milliliter to 1,000-liter reactors, but also from product to product. And what that means is that we can reduce cost, we can keep risks. So this is technical process risks. We can keep them in check and make sure that we deliver on the project timelines, and we can increase model robustness. Now, model robustness is not interesting per se, because there's no value in that. But if you can translate model robustness into process robustness, and you can use that to accelerate development and to take smarter decisions, this is when it really becomes interesting. In particular, accelerated development of biopharmaceutical processes is interesting now in the times of COVID. And I think we all agree that the faster we have a vaccine delivered to as many people as possible, the better our situation will get. In today's talk, I want to start with a quick introduction into hybrid modeling, a rather intuitive one as well. where we potentially will have a lot of engagement. Then go into some research I did when I was still at Newcastle University, which looks at the simultaneous parameter identification and structure discrimination of one particular class of hybrid models. Go a little bit into the dynamic, into the run-to-run optimization with dynamic hybrid models for parallel bioreactors, and then wrap it up with an iterative learning approach where we learn from product to product and can really transfer knowledge across different cell lines. All right, quick introduction. So I typically start with this water tank example, and I like it because it's conceptually simple, but yet it really gives you a direct feeling of how hybrid models work. What we have here is we have this pipe, and on this pipe we have a flow meter, and we have the tank, and via the pipe we fill water into the tank, and we fill the water up to a certain level, and we have a level sensor that tells us what is the height of the water in the tank. And the objective now is that we want to forecast, we want to predict what height of the water level we achieve after a certain time TF, say 10 minutes, 20 minutes, at a constant inflow. So this could be 2 liters per hour or something. Now, if we look at this example, then depending on what your background is, you will solve this challenge completely different. If you present this challenge to a statistician, and this is typically what we see in pharma nowadays, that statisticians work on process development along with the engineers, with process engineers, they will say, okay, we do a series of experiments and we design them in a particular way. And this is what we call design of experiments. So we fix the time because we're interested only at time 10 minutes, 10 hours, right? And we fix the response. The response is really what we're interested in modeling. So this is height of the water tank at this particular time, right? And now what we can do is that we can change the inflow rate. And then in function of the inflow rate, we can predict what will be the height of the water in the tank. So what we need to do is we need to do 3 experiments. And if we do 3 experiments, then we can say whether we will have a nonlinear function or whether the function will be linear. And moreover, we can fit a simple quadratic model to that function, right? So now this function will describe how the height in the water tank will change in function of our inflow rate. So, and this is, of course, for this particular moment of time. Do you think I can still use this model when I change time? Say I'm going from 20 minutes, from 10 minutes, and I want to predict now for 20 minutes. You think I can still use that model? Anyone?
Also darf ich es mal sagen?
Ah, ja.
Ich würde sagen, nein, weil das die Zeit schon fix ist.
Exactly. So we cannot use it because essentially the time is fixed, and therefore, yes, this model can only predict for 10 minutes accurately and not for 20 minutes, right? If we wanted to have 20 minutes as well or 30 minutes, we would essentially have to put in here another term, another domain, where we would essentially vary the time along the. Just one question. I was supposed to talk in English or in German?
I'm sorry, I answered in German. It wasn't, I didn't think about it.
Okay, no worries, no worries.
English is perfectly fine. We want our master students to practice English. And I would say that we also have some people that know German or not so well or don't know German. So English is fine. Thank you.
Perfect, perfect. English is always my first choice. This is, but for a moment I wasn't sure. All right, so exactly. If we have time, then we need to do additional experiments for different time points, and then we can factor time in as an extra factor. So if you come from an engineering background, then at least for me, the first thing I always do is I think about the conservation, and I think about mass conservation and energy conservation, and I will start with material balances, right? So I look how the material changes over time, and the material, of course, here is water. So the density of water can be assumed to be fixed. We know the surface area of our tank, And so the mass is given by the change in the height over time, right? We have D, which indicates the change. This is the change in time. This is the change in material. And because rho and A, they are fixed over time, the only thing that changes is the H. And then the height will change over time in function of the inflow. And here the inflow is liters per minute or liters per hour. In order to have mass, we need to multiply it by the rho. So this is fairly straightforward, right? So if we develop such a model in a process scenario, what we typically do is we do validation experiments. And so we do predictions with the model up front. We predict over time how the height will change. And then we do the experiment here for 10 minutes. And we represent here the experiments by this cross. And as the inflow rate goes up, the height of the water tank goes up, or the height of the water in the water tank goes up. And when you look carefully at the predictions and at the experiments, what can you see?
That they differ slightly. So the experiments also always have a lower height than the predicted values.
Exactly. Very well observed. We have, in fact, a systematic mismatch, right? The experimental values are always below the predicted values. And typically when you have a systematic mismatch, you have failed to account for some phenomena in the process. And here, of course, because we have an open tank, we might have evaporation. And evaporation can then explain for this systematic mismatch because we're always over-predicting. Now, if you want to manufacture evaporation into your model, you need to have an extra term here. And this term will be essentially a function of temperature, pH, and so forth. And it will not be straightforward to model evaporation. In fact, there are many evaporation models out there, and they are fairly complex. However, what we can directly say here is that we can do evaporation specific for the surface area, right? And if we do evaporation specific for the evaporation area, then in principle, we can change the size of the tank and we can still predict for different tank sizes. So, right, if we would now come from an engineering background, we would try to do this model and we would try to model evaporation. But in reality, we're not really interested in evaporation. We're just interested in modeling the water height, right? So what can we do? And this is exactly what we do in hybrid modeling. We use this knowledge which we have, which is the material balances plus this term here. And then we say, okay, this function, we don't really want to develop a mathematical equation for it, but instead we just approximate it with some machine learning method. This could be a fairly linear model or something way more complex, such as a neural network. And here the neural network would be a function of temperature, pressures, and so forth. And the only thing which we'll have to do is a series of experiments such that we can, in fact, identify the parameters and the structure of this function from the experimental data. Now, if I ask you, what happens if I change the inflow rate, for instance, over time? Do you think I can still make accurate predictions with these models if I change the inflow rate over time? So say I've always been operating at 1 liter per hour, and now I'm changing it to 2 liters per hour. Do you think I can still model with this model accurately what will be going on? Or putting it differently, what part do you think of the model would have to be redeveloped in order to account for changes?
I think it should work also for different inflow rates if we assume that our evaporation is not dependent on the inflow rate.
Absolutely right. Absolutely right. So if we assume that our evaporation is not impacted by the inflow rate, right? That means if our machine learning model here doesn't see changes in U, then because we have explicitly accounted for the inflow rate in the mechanistic part of the model, we can make predictions for whatever changes we do in U, and we can predict right. Until, of course, the tank is flowing over, right? This is when the physical phenomena breaks down. This is when you cannot predict anymore. So if I make changes in temperature, say I've always operated around 30 degrees and now I'm going to 40 degrees because it's particularly hot summer, do you think I can still make accurate predictions for that case?
No, because of the term on the second term on the right side of the question.
You mean this term here?
Yeah.
That's absolutely right, right? So this machine learning component, which has temperature as an input, this machine learning component is only good when it predicts in vicinity to the data it was trained on. When it starts to extrapolate, so going away from 30 degrees to 40 degrees, this is when it will likely not predict well anymore, and this means the entire model will not predict well anymore. Totally correct. And essentially, if you look at the two factors that you would have to explore, with U, in the dimension of U, you can go wherever you want, but in the dimension of T, you need to be careful that you, in fact, develop your model in or along these values that you're exploring during prediction. Now, what happens if I change time? So say I go from 10 minutes to 20 minutes. Do you think I can still make predictions with this model?
Assuming that the operation is constant in time, yes.
That is right, right? I can make predictions in time until, of course, this thing is flowing over. So what happens if I change the tank size? I'm going from 1 milliliter to, okay, 1 liter to 10 or 20 liter. Can I still make accurate predictions?
Yes, yes.
Yes, why?
Because only the area changes and the area is not an input in the machine learning model.
Absolutely right, right? We have explicitly accounted for the area in our model. It's not an input to the machine learning model, and therefore we can make predictions for whatever tank size we want. And this is really the strong capability of the hybrid model to make extrapolations. So you can make extrapolations here in dimension, in frequency, in dynamics, and you can also make extrapolations in time without being limited by the amount of data that you have gathered in the past. Now, when we go to hybrid models, they typically consist of two parts. We have a parametric part and a non-parametric part. And the parametric part is typically represented or visualized by a white box, and the non-parametric part is typically represented by a black box. Now, why do I choose the language of parametric and non-parametric modeling? And this is a language which comes from a statistical background. Essentially, what that means is in the parametric component, I fix the structure by knowledge, right? So if we go back here to our machine learning model, to our water tank model, we can see that the structure of this model is fixed from the material balances, right? And this is exactly what this parametric component wants to tell us. The structure of the model is fixed by knowledge. In the non-parametric component, the structure of this model is identified from data or needs to be determined from data. And there is a big advantage. This thing here, essentially, in particular when it comes to neural networks and more complex machine learning models, this essentially can fit pretty much everything as long as there is a systematic trend in the data. Whereas here, you fix somehow the function of the model, and if the data is not meeting your function, you essentially have a problem approximating the data. Now, in hybrid models, we can, of course, combine these in 3 different ways. Either we put the parametric and the non-parametric model completely parallel, or we explore serial setups. We can either put the parametric first or we can put the parametric second. And this really depends on the problem at hand and also on the task at hand. So typically before we develop the hybrid model, we start from what is the objective? What do we want to do with the model? Typically when we develop these serial models, we have a well-structured mechanistic model in place, but then there's some part of this mechanistic model, such as in the case of the water tank, where we don't know what's going on, or we're not fully interested in it, like in the case of evaporation, right? And then in order to describe this part that we don't understand fully or that we are not so much interested in, we essentially plug in a data-driven model, a machine learning model. For the parametric model, this is slightly different. Here, we really have to have a full mechanistic model, and this mechanistic model needs to be able to describe the system. And then the non-parametric model here is just put on top in order to improve the prediction performance of our parametric model. So these models are typically adopted and applied for very different reasons. Many times what we see is these types of models, they are more applied when it comes to monitoring and control applications, whereas these type of models, they are more applied when it comes to understanding and process optimization, process development applications. Also due to the inherent stability issues that you might get when you apply this for process control. Now, let me show you, going away from this very simple water tank example to something more practical, let me show you what really the impact of integrating knowledge into a hybrid model can be. We have here a simple case study. It's Botrytis pertussis. And Botrytis pertussis is essentially a bacterium, and I think it's a bacterium. And this thing here causes whooping cough, and whooping cough is a major reason for death, and you can produce a vaccine, and this vaccine is produced by cultivating the organism. Now this organism, this grows on two substrates, and the two substrates are lactate and glutamate. So essentially this is the food of this organism. And typically when we talk about bioprocesses, we call the organism, or the mass of the organism, we call biomass. What we have here is we have a process where we provide lactate and glutamate in the beginning, and we have some biomass which we put in here, and then we essentially grow the biomass over time. And what this biomass needs is a suitable temperature. It needs to have a suitable pH in the culture, and there needs to be enough oxygen, and this is the dissolved oxygen level. What we can typically measure online, and that is really every 3 seconds, is the dissolved oxygen concentration as well as temperature and pH. So we have a measurement of these every 3 seconds, and typically this process lasts between 12 to 24 hours, depending on what it is used for. In this particular case, we do a batch process. So there's no feeding, there's no additions to the process. And then these compounds here, lactate, glutamate, and biomass, we can only measure them offline. So we have to take a sample, analyze the sample, and then the result will only come back, I don't know, when the process is already finished. So we cannot use those for process control or process optimization purposes, only retrospectively. And we have much less data than we have for these kind of things. So this is our problem at hand, and what we can directly do without generating any data or without looking even at the process is that we can formulate the material balances, right? We see here the change in lactate over time, the change in glutamate over time, the change in biomass over time, and these components, they change, of course, if they react, right? So we see here the reaction of lactate, the reaction of glucose, and the reaction of biomass. Straightforward. Now, if we put a little bit of knowledge inside, and that is really fairly generic still, we can say that lactate will only be consumed if the organism is present, right? So if the organism is not eating lactate, then yeah, there will be no lactate consumption, no change in lactate. And the same is true for glutamate. Now, in case of biomass, we can of course say the same thing, right? If the organism is not present, then it will also not grow. So only if the organism is present, it can also grow. And if we then formulate this little bit of knowledge into a mathematical form, I've done it here in a matrix and vector form, then we can say, okay, if lactate is there, so we multiply with lactate, and if biomass is there, then this growth term which we have here will essentially be non-zero, right? So we multiply lactate, non-zero, with biomass, non-zero, with some specific rates, so to say. We can do the same for glutamate, and then in case of biomass, of course, this is only biomass dependent. And now the problem that we are left with is these rates here and these rates here. So we need to understand how these rates change in function of our temperature and our cultivation conditions. So temperature, dissolved oxygen, and pH. Now, how do we do that? Essentially, we built two different hybrid models here. And we have here, the first one, we only have the material balances, and the second one, where we have a little bit of knowledge inside. We have the same inputs, and this is our online measurements. And we have the same non-parametric model. It's a neural network in this particular case. And this neural network in this case predicts these three rates. And in this case, it predicts these three specific rates, right? And now we've used data of a manufacturing process to fit this black box, right? This non-parametric model and this machine learning component. And what you can see here is the concentration of lactate, glutamate, and biomass over time, the measured concentration, as well as the model prediction without knowledge and the model prediction with knowledge. These experiments here were used for training the model, and these were used for validating the model, okay? So if you look closely at these experiments, what can you see? Or putting it different, is there a huge difference between the experiments? And a huge difference between the modeling performance?
I would say that they perform pretty similarly, but in the bottom part, you can see how they might complement with each other, because the red line is above the data and the blue line kind of trends below. So maybe there's some kind of synergy between them.
You're absolutely right? And they are pretty much the same thing. And here they make slightly different predictions. They could be complementary, but in fact, there's no huge difference, right? And this is, of course, in terms of model performance. In terms of experiments, are the experiments very different to one another? What would you say?
No, not really. I think the trends are pretty similar across the board.
Absolutely right? This is manufacturing data. So in manufacturing, you typically try to have everything as constant as possible, manufacturing in biopharmaceutical industry. Because in fact, your recipe, your process recipe has been filed, it has been approved, and you're not allowed to change it. And because you're not allowed to change it, there's no variation in the data. So in principle, every type of model would somehow learn to predict the scenario well. But what we did now is that we did an extra experiment. And in this extra experiment, we reduced the amount of lactate and glutamate that we have in the beginning of the process. And what you can see now is that the model without knowledge mispredicts both substrates, and it also slightly mispredicts biomass. Now, why does it mispredict substrate? Because it has never seen the substrate concentrations, these low substrate concentrations, right? And it essentially predicts the same kind of profile it predicted before. Now, because we multiply explicitly with the concentration, with the substrate concentrations in the case with knowledge, the model with knowledge, with this lactate and glutamate concentrations, accurately still describes the real process behavior. And this is really the impact of integrating knowledge into the model. That means with the same kind of data, and this is relatively poor data in terms of information quality, we're still capable to derive a fairly predictive model. And if we then generalize these findings, what we can say is that with any kind of machine learning model, if we do experiments here on the continuous black lines for process parameter X over time, then we can, with any type of machine learning models, make interpolations. That means we can, in principle, predict well for everything that is between the things that we tested before. But when it comes to extrapolation scenarios, in particular to range extrapolation, where we go outside of the tested range, With the machine learning model, we will likely not predict well anymore, whereas with the hybrid model, and in particular with the mechanistic knowledge, we can go to extrapolation, we can go to range extrapolation, we can still make accurate model predictions. Now, in case of dimensional extrapolation, it's pretty much the same case, right? We could operate at milliliter and then go to liter scale, and we have seen that in the case of the water tank example. And then we also have the range extrapolation, where we essentially change the dynamics of the process. And if the dynamics of the process are captured in the model, and we have done this, for instance, here because we use dynamic material balances, then we can make good and accurate predictions also if we change the dynamics. Now, as I said, in the case of the water tank, interpolation would be if we change temperature. In the case of range extrapolation, this would essentially mean we changed U here from 1 liter per hour to 2 liters per hour. Dimensional extrapolation would be changing the surface area, so changing temp size, and frequency, so changing the dynamics would be changing U over time, right? We could start with 1 liter per hour and then slowly ramp it up to 2 liters per hour if we start filling in the tank. And still, for all these three cases here, the hybrid model would still predict accurately due to the mechanistic backbone that it has. And this is really the important part, that whenever you develop the model, you need to understand what are the capabilities of the model that are built in. These models do not by accident predict well, right? It's capability that is built in. All right. Now I want to just quickly focus on the other point of hybrid models, and that is essentially the possibility to reduce data requirements. Consider you have a system here with 3 factors. So factor 1, x1, x2, and x3, and we have a response y. This could be the water tank. This here could be time, temperature, and the feed, the water rate. And what we also consider is that there's only main effects and no main effects and interactions, but no nonlinearities. So if we have a machine learning model, we could essentially visualize this by this kind of black box with 3 inputs, 1 output, right? And if we would now have to identify the parameters and the structure of this model, we would have to do 8 experiments. We would have to do on each of these corners here 1 experiment. And essentially, we can represent this by 2 experiments per dimension. And then because we have 3 dimensions, it's to the power of 3, right? Which makes it 8 dimension. Now imagine you in principle know how x3 and some function f, how they impact on the response, right? That means you can represent this here by this white box, which is the mechanistic part. And the only part that you don't understand is how x1 and x2 interact and how they build this function f. Now, in this case, you would only have to do experiments along the dimension of x1 and x2, whereas you could easily change whatever level of x3 you want to have without losing prediction power, right? And essentially, that would mean that you have to do 2 to the power of 2 experiments, meaning 4 experiments in this case. You can also look at the model in a different way. If you would understand how x1 and x2 interact and how they build up this function g, and you would have to then understand how g and x3 impact on y, you would have the same type of scenario. Here, you would have to understand how g, on changes in g and changes in x, impact on y, whereas you describe the changes in x1 and x2 on g by this function here, right, by this function. And that also in this case means that you would only have to do 2 to the power of 2 experiments in order to understand how the system works. So by introducing this knowledge component, you can actively reduce the number of experiments. And here it's a factor 2, right? You will see that when the dimensions increase, and this is particularly also true for bioprocesses where these dimensions are significantly higher, the more knowledge you can bring in, helping you to reduce the number of experiments, the better. Because even with high throughput experimentation, where we have 24 reactors in parallel, it will not be possible to systematically explore in a statistical fashion all kind of combinations that could arise from a theoretical point of view. All right, having finished the first part, which was from a quick introduction, maybe a slightly bigger introduction, but I hope you understood well the basis. I want to go now one step further, and I want to really show you how can we systematically identify the parameters in both parts of the models, and how can we identify the structure, and how can we do that in a simultaneous way. So the way we typically start when we develop a hybrid model is that we set up the model, and then we have this nonlinear or this machine learning part. And in this machine learning part, we have parameters, and we need to identify these parameters. So we have this machine learning part here represented by these nodes connected by arrows. And essentially this node here is some function, could be a tangential hyperbolic or a sigmoid function. And these arrows here, they are weights or parameters. And essentially it's these parameters which we have to identify. So what we do is we use some training data together with some least square algorithms or some other optimization algorithms to identify those weights on the training data. And then we stop the training when we see that the model starts to not only fit the data that we have here for the training, but it starts overfitting data that we have here in the validation. And in fact, it starts fitting the noise that we have in the training. So this is why we have this validation data, which we use for stopping the training. And what we typically do is that we not only train one model, one non-parametric model, but in fact, we train multiple models of different complexity. Because we want to find the model, the non-parametric model, which performs best. So we can systematically vary the complexity of this model, and this is what we typically do. But this also means that we have to train all different kinds of models, right? Which is a lot of time investment. So typically a run on this is an hour or several hours, even depending on how big the data are and how big the size of this model is. Now, once we have trained all these models, we essentially try to figure out which is the best structure. And typically the structure that we prefer, the structure of this model here, is the structure that on the one hand describes our data very well, but on the other hand is as simple as possible, because we want to have as few parameters as possible in the model. And this is all along Occam's razor, which says essentially everything just should be as complex as necessary. And what we do here is that we compute the Bayesian information criteria, and I will show you in a second how this looks like. And what this Bayesian information criteria essentially does, it balances the fit of the model against the complexity of the model. Now, rather than doing this in this stepwise approach and building all these different models, we have asked ourselves, can we do something that we can simultaneously identify the structure and identify the parameters. And what we do is we take a very complex model and we use a regularization technique, and I want to show you in a second what. And with this regularization technique, it is possible to directly put certain parameters to 0, and when they are 0, they are not there anymore. And then at the same time, we identify the parameters. And because these other parameters are set to 0, we also identify the most suitable structure, right? So this is the theoretical idea. So we have shown that this works for one particular class of hybrid models, and this particular class of hybrid models is shown here. So we have here our part that changes over time. And here the right-hand side is composed of two things, and it looks fairly complicated, but it is actually fairly simple. Sorry. This first part here is typically linked directly to the feeding term or the dilution term. And this right-hand part is typically linked to the reaction term, where this is the term, the part that we don't understand, and this is the part that we understand. Now, what we have done in this particular case is that we say, okay, this part that we don't understand, which we typically or traditionally always approximated with a neural network, here, we just use a very complex polynomial function. And this complex polynomial function, in fact, has parameters b, and then it has some mononomials, how we call them. And these things here, they can be components that we think constitute the model. So for instance, we could say, if it was a chemical reaction, we could say we have this concentration terms, 2 different powers, right? This would be a first order reaction, this would be a second order reaction, and so forth. So essentially this here, if you want to say so, is a large library of potential models. Okay, and then the only thing that is missing is that we somehow approximate this guy over here. And how we do that is that we say, okay, essentially we build some kind of approximating function for this derivative. And this approximating function, if we have it, then we can reformulate the problem, okay? So we, for the moment, just assume that this here is some kind of function. Now, if we do this, and we put this guy over here, and we put the terms in here, then this entire thing starts to look fairly complicated. So you can see we have the S term over here, this term stays here, this term is still here, and this F thing is just put in here. And if we then multiply this term on the bottom to the left-hand side, right, and then we subtract, we get this term over here. So this guy looks fairly complicated, but then if you look at it with a different eye, you can directly see the resemblance to a linear regression problem. And this is particularly nice because this problem here is linear in the parameters, and whenever something is linear in the parameters, it's straightforward to optimize. And essentially all these other components here are just x and y's, right? Now we have this linear regression problem. That means we can straightforwardly identify the parameters given that the data is sufficiently rich. And what we do now is that we say, okay, we don't want to solve a simple linear regression problem, but in fact, we want to solve a regression problem where we have a regularization term. And we solve this regression problem by formulating the one norm on the residuals. This is the residuals of our model. And we use the 0 norm on the parameters. Now, what does that mean? So this here is essentially the fit of the model prediction to the experimental values, right? And this guy over here is essentially 1 when the parameter is non-zero and 0 if the parameter is 0. And then the norm says that we sum over all these things. Now, if we formulate the problem in such a way, we can directly solve the problem using mixed integer linear programming. And we can directly put the problem into this mixed integer linear programming solvers. Now, if we do that, then the only thing which still needs to be figured out is how do we choose the appropriate model structure? And here the Bayesian information criteria comes in. We have here the fit of the model and here the complexity of the model. So df is the degrees of freedom. And so this BIC essentially balances the fit of the model against the complexity of the model. So what we do is we vary the gamma, which is the regularization parameter, and then we compute the BIC for different gammas. And typically what we see is that we obtain some kind of function and we choose the BIC with the highest value, because essentially that would mean that this structure is the best structure because it produces a fairly good fit at fairly low cost. Cost meaning a low number of parameters. So how does our approach look? We start with the experimental data, and from the experimental data, we then use some spline function to estimate the derivatives. With the estimated derivatives, we go into this MILP approach and we optimize the structure and the parameter at the same time. We then put the structure and the parameters forward. We simulate the optimal model, which is coming from here. And we simulate in an ODE fashion and integrate the problem. And here we do a re-optimization because we want to make sure that the parameter estimates that we have are statistically optimal. And then once we have done that, we can see whether the measure, whether the goodness of fit is sufficient. Then we have our optimized model. And if it is not sufficient, we can iterate until we are happy with the simulated system. Again, we'll pick a simple bio case here. It's a simulated case because for these kind of problems, we're always working on simulated cases. And in the simulated case, we have a change in biomass. This here is the dilution term. So when you feed, essentially a biomass is diluted. This is the substrate. So substrate is consumed in function of biomass growth at a yield factor y is x. And a substrate also changes, of course, in function of dilution, but also we feed substrate over time. So we add substrate, right? And then volume changes in function of the feed. And we assume here that, we can neglect the rho. And in this simulation case, so this is the case that we use to generate the data, this growth function here is essentially a function of our substrate. That means at the beginning, as substrate concentrations are low, you will increase almost linearly with increasing amount of substrate, but then at some point it will flatten, right? So the curve looks fairly like this. Now, if we use this function, then we can generate experimental data, and we have used here two different feeding rates for generating this data. And this is particularly important because otherwise the data is not sufficiently rich in information. At this first instance, we have used an exponential function, and at this second instance, we have used a linear function. So the feed, this thing over here and this thing which goes in here, is essentially changing, and therefore the dynamics of the system are changing. And then we have simulated that we can only take measurements of biomass, substrate, and volume at discrete moments of time, as it is in reality. In fact, we have assumed here that they can be fairly frequently measured, so I think every 1 or 2 hours. Now we've come up with our hybrid model structure. So this was the structure which we used to generate the data, right? Which in reality would be the real process. And now we have this model which we use to model the process in reality. Here we have our general structure, and we came up with 2 models here. Okay. In the first model, maybe we start this way. In the first model, we essentially reformulated the balances to really make it material balances. And you can see here the change of mass, of biomass. This is the biomass concentration times volume, which is changing in function, of course, of biomass itself. Biomass only grows if it is present. And this is somehow the growth rate. And then on the second part, we reformulated the substrate balance also again to make it mass here. And essentially this is the feeding you're putting into the reactor. So we put that on the left-hand side because we know all these parts. And then on the right-hand side, biomass is only consumed if, substrate is only consumed if biomass is in the reactor. And this here is somehow, again, an approximation of biomass, even of the biomass growth rate, even so we don't know that it is biomass growth rate. Now, in the first case, we have directly used the material balances as we had them before. And essentially, we tried to estimate here this f functions, and this g1 functions here is the multiplication with biomass. And in this case, it's the multiplication of biomass with y is x. Now you might say, okay, why do you do 2 times the model with almost identical knowledge? Now the difference is that in this first case, this left-hand side is change in concentration. So the spline approximates change in concentration, whereas in the second case, the spline approximates change in mass. And you will see in a second why that is important. So what we have done is for different penalty parameters, for different regularization parameters, we've estimated our parameters, which directly gives rise also to the structure, right? And then as we increase the regularization parameters, the model parameters drop out. You can see here some of the model parameters are taken to 0. And then at some point, there's only one model parameter which is remaining. And if we look at the BIC and the degrees of freedom, you can see that the number of degrees of freedom, this is really the number of parameters, drops as we increase the regularization term, which is what we would expect. And you can see here that the BIC, which indicates the most appropriate model structure, is the highest, in fact, when we are at a high regularization parameter. And so that indicates that the model that we are looking for only has one parameter. Now, the interesting thing is, we can do this with both models, and so with both hybrid models. And in fact, the model comes up with the same structure. But the parameters in the model, they're different, right? So in this case, it's 0.48 and 0.39. In this case, it's 0.40 and 0.51. And this is a significant difference in terms of biomass performance, right? This is almost 10% more if you want to think, though, 20%. So in reality, this is the original model. You can see that in reality, it's much closer to 0.4 than it is to 0.5, right? Now, why do the models perform so differently? The difference here is really the data, the approximation. So here on the left, we see the splines, which are approximating concentrations. So in red, you can see the true specific growth rate. And in blue, you can see the spline approximated specific growth rate. Why is it so bumpy? Because first, there's only a few number of measurements that we have available. And second, there's noise on those measurements. And because building the derivative is always a mathematically ill-posed problem, noise typically amplifies, and therefore we get these bumps in this function. Now, if we look at the derivative that we built on mass, so the spline on the mass function, then you can see that this one is much smoother. And because this spline here is much smoother, it also introduces less errors into the optimization that we do on the parameters in the structure. And therefore, essentially, the model parameters on the second part are much better than on the first model. And this is also according to observations that have been reported elsewhere. Now, let me finish on this first part where we looked into a fairly complex parameter identification and structure discrimination problem. And we have shown that for this particular class of hybrid model, it is possible to, at the same time, identify the parameters and discriminate the structure. We can solve this problem in significantly less time, so almost on a run, than with standard approaches. And it has been shown, for instance, for plug flow reactors that this can directly be applied. And then one thing that needs to be said is that the formulation of the modeling problem, and this is what we have shown with model 1, model 2, can have a significant impact on the parameter estimation performance. And this is something we see in general, no matter what type of hybrid modeling. So when putting up your modeling structure, thinking about the parameter identification and the structure discrimination part is always an important component. If you're interested to know more details, you can find more details in this publication, which is from the time that I was still working at Newcastle University. All right, if we look generally, and I just want to point out some challenges that we see when it comes to parameter identification and structure discrimination in dynamic problems, we have this bias variance problem. And bias really means the kind of functional the kind of function that your mechanistic model produces, and that somehow hinders your system to really approximate the data. And for this mechanistic part, we typically look into identifiability. And in fact, when we look into the hybrid modeling literature, there's almost no systematic evaluation on identifiability and inductive bias. And at the same time, we look at fitting the variance. So we try to approximate the remaining part of the model with the machine learning component. And here there's very little work done on overfitting. In fact, most of the approaches that we're still using in the hybrid modeling literature stem from the 1990s. And so they're fairly old. And I think there is a significant room for improvement here. Then generally, when we look at parameter identification, not only the non-parametric, so the machine learning part can have parameters, but in fact both models. And we could go even one step further when we look into symbolic regression, where we try to really identify the true mechanistic model from data, and where we could also integrate what we already know about the system. And then when it comes to learning, and this is particularly also interesting with the next problem that I will show you, what we do typically nowadays is batch learning, meaning we start with a certain amount of data and we try to identify what's going on. But as with processes, the data size grows, we cannot always retrain the model because it becomes computational too expensive. So we need to understand how can we only use the newest data to retrain our model and to take already forward what we learned about the system at hand. All right, let me quickly go into the second part, and I'm fairly aware that I'm already talking for a very long time. So the second part is really the interesting part. this is where the added value is generated, so to say. What we typically have here, and this is directly based on my practical experience in industry, is that we start from some statistical experiment design. We do some experiments on a high throughput platform, and then we take the data forward, we train the model, and then we design the next round of experiments with this model. And this is straightforward when this is only a few reactors in parallel, like 1, 2, 3, 4 experiments. But if we have, with modern approaches, 24, 48 reactors, and you want to design in parallel 48 experiments, and in particular when it comes to designing them on the fly, things become fairly complex. So what we're looking at here is a case study which is fairly similar to the ones before. Again, it's a bioprocess problem. In this case, we call it a Chinese hamster overall organism. It's a fed batch process. We're producing a monoclonal antibody. We can measure viable cell density, glucose, glutamine, ammonia, lactate, and titer. We're optimizing here temperature, pH, feeding, and the stirrer speed. We have four parallel reactors for the sake of simplicity. And we assume that at the beginning, we only have data from 4 reactors available. Now, what we can do here is that we essentially use the same kind of model. We have here, again, the hybrid model, which is the machine learning, the material balances. And then in the material balances, we have this reaction kinetics. And then these reaction kinetics, or the specific rates, they're essentially modeled by some machine learning component. Here we use artificial neural network, and this artificial neural network is essentially fed with pH, temperature, and so forth. And now is the interesting part. We can come up with different strategies, and I don't want to go too much into detail. We can come up with different strategies how to design the different reactors. So in this first case here, and I only want to focus on strategy 1 really. In this first case here, we maximize the product. This is titer. We maximize titer at the end of the experiment. And then in the follow-up experiments, so experiments 2, 3, 4, which are running in parallel, right? We want to maximize titer, but as we maximize titer, we also want to do something else. And this is represented by this very complex function. And this very complex function looks only very complex. In fact, it's a sigmoid function. So sigmoid function is 0. And so on the y-axis, it's 0. And then when it comes to where the x-axis is 0, the sigmoid function goes up to 1, right? So essentially, if we look here, we have the titer that we maximized. And this is the titer. Oh, there's an indice missing. This is the titer we're maximizing here. And then we multiply it with some r, which is essentially a risk. And then we look at the variation. And this is the prediction error, so to say, the prediction of our model. And what we do here is that we say the prediction error, so the confidence interval, cannot be larger than the predicted titer times some risk measure. So typically we can use here values like 30%, 50%, and so forth. And then depending on how much uncertainty we will to take, we essentially constrain our optimization approach. Now, in this second strategy, we do a fairly similar design where we essentially maximize the same function as here, and then we want to explore the space very efficiently. In this strategy over here, we again maximize titer, but then we explore the space in a slightly different way. And then in the sixth strategy here, we're really into maximizing expected improvement. So we want to look into process points into where we can learn the most, and which at the same time somehow are very likely to optimize titer, to maximize titer. Now, if we do these experiments, remember, we're always doing 4 experiments here in parallel. We start all from the same model. And then the model is predicting for the next 4 experiments what will be the titer, right? And you can see here is the prediction in blue and the confidence bound. And you can see that it makes fairly high predictions. And then if we do the experiments, you can see that the experimental points are fairly low. And this is what we typically see in the beginning, because of course, the model was not trained on data where it's predicting. And then again, we use the new data. So this is then 8 data points to train the model, make new predictions. do new experiments. And then as we go on, you can see that titer improves, and also the prediction, having very large confidence interval in the beginning, get better and better. You can see that in case of strategy 1, this works really well. For the other strategies, the error bounds are fairly large, and somehow the experiments are scattered all over. And knowing that the theoretical maximum point is at 1,000 milligrams per liter, you can see that while strategy 1 almost gets there, none of the approaches really gets there. And, so what we are interested at the moment in is to explore optimization criteria that can more efficiently learn and that get better to the process optimum. What also needs to be said is that this here is just a representative example. In order to really make it statistically significant, you have to rerun this thing several thousand times, and this is what's still running while we're speaking. So already wrapping up on this part, and this is before I just want to dive in the last part, we can say that currently the performance is not fully satisfactory, as I said, where with strategy 1, we seem to be converging mostly and most frequently towards the optimum, but we're not fully getting there. And then we're also looking, and this is something where Arthur has published as well, into more orthogonal measures for evaluating the risk of misprediction. And we hope that these kind of measures could help us to exploit the process space more effectively. Now, looking a little bit ahead, what we have essentially done is we've run this thing in parallel, and for a single reactor, we have this already established. It's called run-to-run optimization, but there are several different problems that are super interesting. One thing is doing it sample to sample. So that means you're really optimizing your process on the fly while the process is running. You're re-optimizing it. You're changing the set points. So this is super interesting, an area which is also super interesting to work on and to investigate. And the other area which is super interesting as well is calibration design. And this is really given that you have some data of similar processes of the past, or you have some model of the past. How can you design calibration experiments that are such that you can then use some background knowledge to iterate faster here, right? So to learn faster, more effectively, and to do less experiments from the very beginning. And this is something where we're working on as well. Because essentially what we're doing nowadays, if you look here at different drug candidates, so this could be drug 1, drug 2, drug 3. At the moment, if you develop a process for this new drug, you almost take every time the same number of experiments. And this here is thousands of experiments typically, right? It depends a little bit on the platform. So this means 2 years of working time. Typically, we don't have the 2 years. So we want to go lower. And what we have shown is that with these kind of hybrid modeling approaches, we can reduce the number of experiments. But we're still somehow developing the process de novo every single time. Where we ultimately want to go is that we want to reduce the number of experiments iteratively so that in the future, we could think 5 years ahead, there's 2 or 3 experiments that we need to do. And we are then have our process fully developed, just based on a computational approach. That's really the aim. And this is where we're aiming at DataHow. And this was also the aim of my group at GSK. And here I want to show you first step towards this. So what we have done is the same model as you have seen before. It's not exactly the same, but conceptually it's very similar. And here we have a difference. So here, when we go from one product to the next, we let the model know that we changed the product. And when we change the product, we also call this cell line, because essentially when you change the product, you also change the organism which is producing this, or you change one part of the organism, so to say. Now, in this particular case, we'll look at 5 different products, and these 5 products have the same kind of process parameters. So this is temperature, feeding rate, and so forth. But the products are also different. So how do we represent to the machine learning models that the products are different? Now, what you typically can do is you can say, okay, product 1 is 1, 0, 0, 0, 0. Product 2 is 0, 1, 0, 0, 0. And then you get the drift, 0, 0, 1, 0, 0, and so forth, right? So this is what we call one-hot encoding. The problem is when you have a 6th product down here, because in the past you don't have data of this product, you cannot make any predictions, and it will take you some time before you can build up how the model will look for the sixth product. Now, what we do here instead is that we say, okay, we go to some embedding space. And this embedding space is somehow a reduced space where we capture the similarity of the products. And essentially the dimensions or the coordinate system, so to say, of this space is described here by this capital letters E. So we have the first axis, the second axis, and the third axis, and then the position, the relative position of this product here in this space is represented here by A1, B1, C1, and then A2 and so forth in the other ones. So what we now need to learn from the data is first we need to learn the coordinate system, and second we need to learn for every product where in this coordinate system it comes to lie. Okay, and essentially these things we call here embeddings, and this is the position in the embedding space, so to speak. So the first thing we need to figure out is what is the dimension of the space. So we have developed models for several dimensions, dimension 1 to dimension 5. And here you can see the error in prediction for our product. And you can see that the error in prediction goes down until dimension 3, and then it stays fairly constant. And also, if we look at the log marginal likelihood, it increases all the way to the third dimension, and then it stays fairly flat. And this really indicates that 3 dimensions are sufficient here for our 5 products in order to capture the similarity between the different products. Now, when we then go and we want to make predictions for a new product, we essentially look here how many new experiments, how many data of new experiments we need in order to become predictive for this new product, right? So we can see here 2 new experiments, 4 new experiments, 6 new experiments, and 8 new experiments. And we use now this model that we have trained on the different historical data with the embedding, and we start making predictions if we only knew 2 new process data runs available. And we compare the performance here to this one hot encoding. This was this 1000 and 0100 and so forth. And we also compare it to a model that you just train on this 2 new data points, 2 new experiments, sorry. And okay, for 2 new experiments, it's not so interesting, but if we then go to 4 or to 6, you can see that the model that has taken historical information into account is significantly better than the model which only takes the current experiment into account or which uses the traditional one hot encoding. And why is that so interesting? Because essentially, starting with 4 experiments and then going to 8, with 4 experiments, you're almost as good as with traditionally 8 experiments. And then with 8 experiments here, you're almost as good as traditionally with 16 experiments. What you can say is that CS at the limit, so as you go to more experimental data, the model that you just develop on this new experimental data is capable to describe this local product, this one product, much better than the behavior of the system of this one product better, but it is not capable to describe the behavior of all the other processes. And so what we now have is a model that really for very few new data can directly make predictions, and we can directly use this model to describe the system behavior of a potentially new process. And this is super interesting. And what is even more interesting is this position in the new coordinate system, right? So we have here our traditional products, and we have here our new product. And what I can say about the relative position is that these guys over here, they are consuming a second substrate, which is lactate, and they start consuming this at some point, whereas these guys over here, they do not consume this. So somehow, implicitly, not only are we capable to predict better, but we also generate some insight into the process dynamics. And this is for a process engineer really, really, really interesting. So, yes.
Sorry. We got another few minutes.
Exactly. I'm wrapping up, and then
thank you.
I would jump into questions. So essentially, what I have hoped to show you is that with this embedding technology, we can really describe process data across several different products. We can provide information how similar the products are relative to each other. And what we hope to do in the future is that we can make process development significantly faster. And these results have just been well, submitted to the biotechnology and bioengineering journals, and the preprints are available here. Wrapping up, I just want to say that we should give a much better appreciation to prior knowledge, and we should start integrating it more and more into our modeling activities. And in particular nowadays, where the entire world is focusing on machine learning methods, we should make sure that we're integrating this along with machine learning, because otherwise we will learn everything from the scratch, and most of the stuff we already know. And the other point I want to make is that when we look at the challenges that lie ahead, and that is really on-the-fly optimization, optimization of parallel reactors, and across product optimization, in the area of bioprocess optimization, there's almost nothing published there. There's almost no one working there. So I highly urge you, if you're interested in this area, to go there and do something. I want to finish with some acknowledgements, and this is also for the reading list. I want to thank Nico, Ale, and in particular Clemens, who was working on this knowledge transfer part, because these guys have been working hard, and this is something which I particularly love in DataHow. We have really fruitful brainstormings, and this new and different ways of looking at problems is always highly engaging. And then I want to thank Mark Willis, with whom I worked a lot at Newcastle University, and with him together I worked on the simultaneous parameter identification and structure discrimination problem. And yes, also with him I had a really good, and I still have a working relationship. With this, I'd like to thank you for your attention. I've taken slightly longer than I expected, but I'm happy to take your questions.